Hello, this is Dr. Dan Guerra coming to you from Authentic Biochemistry Studios in the beautiful inland Pacific Northwest. Today, I believe, is the 6th of May, 2020. I believe it because I have coherent and foundational evidence that is true. So let's just go with that and move right into this lecture. So let's do a slideshow. From the current slide. Okay, so I'm going to be continuing along this discussion of immunoendocrinology disease. This is actually the 9th episode, and I'm going to be bringing back my favorite topic, fatty acids. There's not going to be that much information directly about lipids right now, but I'm going to introduce it, and it's going to actually appear to some to be a main theme of the talk, but it's really only going to be sort of peripheral to drive home some important aspects of how intermediary metabolism controls hormonal regulation, and hormonal regulation responds by mediating intermediary metabolism in different cells, tissues, organs, etc., to generate finally a homeostasis even upon stress in the human body. Since we're talking about disease, we want to know how disease can be afforded even when there's a normal challenge of, say, etiologic agents like bacterial infections or fungal or parasitic or viral infections. How is it that some people get really sick? Other people don't seem to be very sick at all. Obviously, it's the immune response, and we're going to try to trigger back into the immune system so I can generate my diet event ontological perspective at the end of this long arc of lectures, which I most certainly will do. So, as you can see today, I have an image of myself over here. It looks just like I do because I just took that picture. So, a lot of the pictures I have were taken different times, usually right around the same time. I try not to take anything from really when I was younger because I looked a lot different and not nearly as like normal as I do now, or non-normal as I do now. So that's a picture I just took. So anyway, there you go. All right, so let's get started. Now, on the audio discussions I was having on the podcast, Authentic Biochemistry podcast, this is a production of Authentic Biochemistry, of course. I was looking at this paper towards the end. Journal of Physiological Biochemistry, published not even a year ago. They looked at mRNA, transcription that is, protein secretion, and CpG methylation as an epigenetic marker of the following inflammatory genes: IL-1 beta, TNF, plasminogen activator inhibitor 1, which is also known as SERPINE1, and interleukin 18. Treatment with palmitic acid increased 2 of those pro-inflammatory cytokines. Contrary-wise, that's my term, the supplementation with docosahexaenoic acid, polyunsaturated omega-3 fatty acid, found in the diet, but also as a supplement, reduced IL-18, TNF-alpha, and PAI-1. Okay, so all 3 of those potentially pro-inflammatory cytokines were turned down by just the manipulation of fatty acids. So they say that incubation of these fatty acids did not modify DNA methylation. So they did a DNA methylation, a methylomic study for the enhancer regions or the chromatin near the promoter regions that have an enrichment of CpG islands, wherein the C5, or the cytosine residue of that CpG island, can become methylated. I mentioned this to you in the audio. I just want to mention it to you in the video. I've done these kinds of experiments, and basically what it is subtraction. And you make a primer and run PCR, of course, and you're looking for something you're priming that is in that CpG island rich enhancer region or promoter region of the gene you're looking at. So you have to have flanking sequences so that you know you're actually looking at that gene, right? And it's kind of by hit or miss if you know whether or not you're looking at the correct or appropriate cytosine P guanosine rich island, because those are the areas where often you get a methylation hit, that its methylation is increased. And to remind everyone, the higher the level of methylation on some promoter regions, the less you're going to get transcription. So hypermethylated DNA tends to be lower transcribed than hypo or non-methylated. Not always the case, but that's the standard paradigm. So it's not absolutely clear when you make the primers for this PCR reaction, when you're looking to see basically if it's protected, because what you do is you use a system where if it's not methylated, the DNA is not methylated, then that region of the DNA is lost, okay? So that means, obviously, that if you can prime it, then that means you have a methylation, an area that has been methylated, okay? So that's what I'm talking about. So there's a destructive technique you use even to be able to probe this pathway. All right, so having said all that, it is pretty clear, though, when you have a strand of DNA that is methylated upon some kind of induction, like a stress induction, especially when you're doing an animal model, right? You're going to be looking at, for example, we were looking at the brains, regions of the brain, particular nuclei. In fact, similar ones that are talked about in these papers, hypothalamus, pituitary. We also were looking at the prefrontal cortex and the medulla, and also other regions associated with affective neuroscience. So what I want to say is this. The technique of studying methylation has been worked out for the last 20 years, and it's pretty rock solid. If you have the right region of DNA that you are trying to expose as being methylated or not, based upon some treatment, like here they're treating with fatty acid. But if you miss the mark, if you don't get the right region, you don't have enough time in the day or enough money in the grant to be able to look at all the potential CpG sites. So it is I wouldn't say it's completely stochastic how you choose your CpG islands, because there are programs that help you select them. But the programs are simply based on, does the literature say these regions are normally methylated or not? A, or does this constitute a super high abundant level of CpG iterative occurrence, which is what you mean by CpG island, in the promoter region or an enhancer region of a particular gene. Neither one of those is absolutely necessary or universal. Two words are supposed to be giving you an absolutism in science, which of course, I don't feel at all possible. But taking that into consideration, there's no way of really knowing you're looking at the right CpG island. This kind of work usually has to be backed up with doing more analysis of CpG regions all around a promoter region of a specific gene, determining which ones may or may not be methylated and which ones remain methylated. Right? So maintenance methylation is just as important as episodic methylation. Yet episodic methylation can alter gene expression. That's how epigenetics works. It's a difficult thing to track. All right, so having said all that, they didn't find any methylation pattern. So they say the fatty acids weren't working at the epigenetic level. They say that that palmitic acid induces important pro-inflammatory markers in human macrophages, whereas DHA decreased them. And they're saying apparently methylation is not directly involved in this. Now, I would say, because of what I just described to you, they should have done a better backup here. So I won't say that it's just a terrible paper, but I'd say that there is a poor design of the experiments, because they don't recognize, forget about what I just said, bracket that off, about getting the right CpG target, but they weren't looking at any other potential epigenetic phenomena. And that's what this paper was looking for. Is there an epigenetic phenomenon? Is fatty acid acting as an epigenetic agent, delivering an event which results in a methylation pattern, which turns down expression of genes? For example, DHA turns down expression of these pro-inflammatory cytokines. So that was what they wanted to look for. They were trying to look for evidence. They only look at methylation patterns. What about acetylation of histones? What about acetylation of DNA? What about microRNA? Those are all composite, powerful components of epigenetic control of the gene expression. We're looking at them. So I would say that I would give this paper, if I were to have reviewed it, I might have accepted it, but I would have asked them whether or not they had any other inclusive data looking at other possible routes of epigenetic alteration of gene expression. They didn't. I would say probably the paper could be accepted because there's a lot of papers that come through this way, but that I wouldn't probably refer to it very often. Now, the only reason I'm bringing it up now is not because I think it's such a great paper, but because this is very common in the scientific literature. You get a paper published, you get a very bold statement that says, well, we looked at fatty acids and wow, look at this saturated fatty acid, all this effect on pro-inflammation, right, in macrophages, for example. Wow, so it must be the turning point of macrophages. They become pro-inflammatory if they have saturated fatty acids. And then vis-a-vis the diet high in saturated fatty acids. And no way at all did they demonstrate this. For one thing, and I said this in the previous talks, you synthesize palmitic acid de novo from fatty acid synthesis. Common fatty acid in membrane lipids as well, number one. Number two, you also produce, or after the ingestion of essential omega-3 fatty acid, alpha-linolenic acid, or directly in the diet as DHA, or even EPA after it's been converted, you have enough DHA in your body that you would be thinking that there would be a rheostat control, a teeter-totter control of the amount of palmitate versus long-chain polyunsaturated fatty acids of whatever positional series of desaturation they happen to be, omega-9, omega-6, omega-3. So that's the point I'm making, and that's the problem when people do work with lipids and they don't consider all the lipid pathways. They just keep on bringing that home because lipids are so essential to understand disease in humans. And yet, there are very few people that are really using the powerful, not so much analytical tools, not so much just purely empirical consideration of science, but the reasoning aspect of science, the conceptual framework and the ideation that goes around just plenty of experiments. And for that, you need a lipid biochemist. That's the point I'm trying to make. And if it was somebody that was if you're looking at nucleic acids, you should have a nucleic acid biochemist on the team, maybe helping you design those experiments. Likewise, carbohydrates. Who would want to work with carbohydrates? I don't know. But there always are people. It's supposed to be a joke. All right, now, here's just a taste of what you might get from fatty acids that have nothing to do with producing ceramide. Remember, we have been talking about ceramide, which uses palmitoyl-CoA in the de novo pathway, the serine palmitoyltransferase enzyme. And that's how you make the backbone sphingosine, which ultimately another fatty acid is loaded to the nitrogen atom previously on the serine amino acid. And that amide linked then ceramide after, of course, the desaturation of the palmitate, that trans configuration in the backbone. You get the production of any number of series of ceramide molecular species based on what the composition of the fatty acid is linked to the nitrogen atom. And I told you some of those ceramides can cause lipid raft migration in and out of the plasma membrane, in and out of the ER, mitochondrial membranes, even towards the nucleus, certainly the Golgi and endosomal complexes. And that then causes either an aggregation or a dissimilation of receptors on the surface of the plasma membrane that can then allow for things like immune cells to operate, or even foreign etiologic agents like bacteria or viruses to enter into a cell and then cause a pathogenic invasion. Right? We talked a lot about that in these 8 previous lectures leading up to this, and then before that, many more. So I want you to make that clear why ceramide is important. But palmitate can also be used directly to covalently modify proteins, and that's what this slide is showing you. Not only palmitate, right, but you can also get farnesyl groups added to proteins, geranyl. These are prenolipids, and you have multiple ways of prenolating proteins. You can myristate. That's a C14 saturated fatty acid. Sometimes it's hydroxymyristate. Okay, a modification even of the C14 saturated fatty acid, and that is a co-translational phenomenon. It hooks right into the translational machinery of certain proteins made, even in polyribosomes in the cytoplasm. That's right. Then you have all these palmitoylations. Okay, so you take a look here. This creature here. So you have multiple ways of adding palmitic acid. And all of these different introductions of palmitate are sometimes on different amino acids in a protein, endo versus exo. Most of this is occurring, well, I shouldn't say most of it. It's pretty much stochastic throughout the whatever protein is becoming palmitoylated. And when you palmitylate a protein, you also cause it to migrate. Migrate where? To some places more hydrophobic. Like what? Like the plasma membrane or intermembranous system. See? So what I say here is protein lipidation invents cell responses to extrinsic and intrinsic environmental stresses. So getting that covalent modification of proteins can also be triggered by an immune response, or it can facilitate immune response being triggered. Okay, so that's what I mean by stresses, internal, external. It doesn't matter. Overproduction of certain proteins in cells, the entire apoptotic pathways or necrototic pathways can be induced by prenylation or acylation via palmitate, myristate, stearate, any fatty acid you might consider that would be nearby as a CoA thioester and associated with a specific lineage of mechanisms leading to a change in the fate of a cell based on some kind of pathology that's associated with or without a meteorologic agent, exogenous or endogenous. Okay, so by that means basically cancer can also cause this, or just a chronic disability like obesity or aging. Aging and chronic disability, right? Yeah. Okay, by modulating protein hydrophobicity, so what's going on here? You're modulating protein hydrophobicity by adding these lipids, stability, and basically that biophysical association like grafting and whatnot. Plus changes in pathway dynamics also get altered tremendously because of this. Enzyme kinetics, Km, the Vmax of enzymes, cell fate, as I've been talking about, and then an alteration completely at transcription level, gene expression, or even at translational or post-translational modifications of proteins can be mediated by the acylation or prenylation of proteins involved in those pathways. Okay, and so that all over the map, transcriptional, translational, they weren't looking at any of that in the paper we just talked about. I'm not saying they have to look at it, but the fact that they didn't and didn't mention it, these are modifications that can alter gene expression. And the palmitic acid, if it is involved, may be working at a different level of molarity even than the DHA. Right? So these are the really fine-tuned experiments. You might think this is really getting down into the molecular level. Well, that's right. It is at the molecular level. So that's exactly where you need to be if you're a pharmaceutical scientist, or if you're a biochemist, or if you're a cell physiologist, or if you're an oncologist or cardiovascular surgeon. You need to have some understanding of down to the molecular level. You can't paint broad strokes by saying high-fat diet or saturated versus unsaturated fatty acids. For all the reasons I'm trying to key into you, because what happens in the body at the very specific molecular level is occurring at a subcellular level as well, and different cell types, different tissue beds. You know what I'm saying? All right. So an increase of histone acetylation has been reported to positively correlate to the production of cytosolic acetyl-CoA via the ATP citrate lyase pathway. Okay? That suggests anabolic status of the cell links with euchromatin maintenance and an uptick in global transcription. I got that from a science paper published 11 years ago. Another important thing I say in Authentic Biochemistry. Papers that are published not just in the last couple, 3-5 years matter. Papers that were published in any decade, you have to know what to ask for in the scientific literature. It is a very large corpus of information. It's not all what I would call knowledge, but it's information with data. And that data then has to be transcribed, if you will, by someone with the understanding of how data can be transcribed, that is a human mind, not a computer algorithm, to be able to know whether or not that data turns into evidence for something. And then ultimately, the human mind has to work to determine if you can verify the evidence. So there are multiple levels there too, even when you're looking at scientific literature. How do you know that that literature is good literature? Just now, like what I was talking about, that previous paper. Again, the paper is not a terrible one, but it makes a broad generalization. It gets picked up then by any other number of laboratories, and then they run with that. Well, there's no methylation pattern here. Or they try to say, well, let's see if they're right. And if they don't find any conflicting evidence, and there it sits in the literature, then it becomes a paradigm. And then it becomes dogma. And then people start saying, well, there was no evidence of that. Yeah, well, was the experiment done correctly? You get the point. And you know how many papers are published every day in multiple journals, and it just keeps on increasing. And the older work cannot be discounted. That's what I'm saying. Look at this. Acetyl-CoA via ATP citrate lyase. That embodies a whole understanding of the TCA cycle. The fact that there's plenty of NADH in the system, either coming from the cytosol via translocation of the reducing equivalents I talked about, or directly from the glycerol-3-phosphate shuttle, and then NADH being generated from glycolysis or pyruvate dehydrogenase reaction, for example, running carbohydrate through there, or all the NADH is going to get made from beta oxidation of fatty acids directly in situ in the mitochondrial matrix. When you make a lot of NADH, you block all the dehydrogenases in the citric acid cycle, and so therefore citrate leaves the mitochondria, and that cell becomes anabolic, and the first thing it needs to make, because it can be readying for cell division, is membrane. In order to make membrane, you need lipid, so you need de novo fatty acid synthesis and de novo cholesterologenesis. And both of those pathways start with acetyl-CoA cytosolic, and they all come from this citrate lyase pathway. A couple of other ways to get carbon out there at the acetyl-CoA level. The only other one of really significance that could make lipids, though, because remember, fatty acid synthesis occurring in the cytosol in animal cells, is by the utilization of ketone bodies, which ketone bodies, of course, were generated previously, or that is ultimately, if you want to look at it that way, by some cells' utilization of preformed fatty acids generating acetyl-CoA. Then acetyl-CoA then being converted to acetoacetate or beta-hydroxybutyrate, ketone bodies being sent somewhere to another cell tissue, and then ultimately you can reform the acetyl-CoA and use it to make fatty acids in an adjacent cell. Of course, why not? And now you're back on the way of making palmitate, or stearate, or even oleate. There you go. So, would expect this state in organs, tissues, or cells with de novo lipogenesis or ketone body utilization is active. That is, you would get a lot of this ATP citrate lyase activation. Besides the adipose, skeletal muscle, liver, kidney, and cardiovasculature, of course, the CNS is also very active in de novo lipogenesis to serve the turnover of synaptic junctions and axonal myelination. That's really important in the CNS. You make a lot of fatty acid, and you elongate and desaturate a lot of fatty acid directly in situ in the brain. So murine maternal high-fat diet, it goes back to an experiment taught using HFDs. Murine, you got a mouse model, not a human, was associated with reduced SIRT1 expression. Now that's a deacetylase. Okay? That is a histone deacetylase, or an HDAC. Okay, so you get a reduction of SIRT1 expression. So if you get a reduction of that, what happens in the hypothalamus? It also is occurring in white adipose tissues in the liver. So let's look at what happens if you overexpress the SIRT1. In those offspring, you get a significantly attenuated excessive accumulation of epididymal retroperitoneal white adipose tissue. Okay, so you attenuate that. So that means that what? So overexpression of SIRT1, let's look here again, in these offspring, significantly cut the excessive accumulation of white adipose tissue. So that means that deacetylation of whatever the substrate is for that enzyme, it could be histone, is going to alter gene expression, and that makes it an epigenetic phenomenon, right? Directly. Now, is a high-fat diet directly involved in that? Well, that's what they were looking at. But the high-fat diet doesn't just make available certain fatty acids. It doesn't just turn on a particular deacetylase. It tremendously affects the entire homeostasis of all the metabolic pathways which are networked and put into one grid, which is the homeostasis, and the procurement of survival and existence of cells, tissues, organs in the human, which is what we're most interested in biomedicine, at the individual level. All right. Okay, so this gets us this chapter in this book, published about 10 years ago, hypothalamic fatty acid sensing in normal and disease state. You can see the authors. Hypothalamic fatty acid levels and metabolism have been shown to regulate both food intake and exercise. Hyperphagia, overeating, and obesity resulted after the ventromedial nucleus, VMH, of the hypothalamus, the VMH, was subjected to bilateral lesions or following administration of the non-metabolizable 2-deoxyglucose. In other words, starting glycolysis but not able to finish it, not able to run any carbon through it. And that was into the third ventricle of the brain. The hypothalamus has nuclei that are interconnected, of course, via axonal projections, equipped to respond to environmental cues linked to stress, immune responses, and bioenergetics, such as nutritional addition. And these are all considered as rheostatic controls involving the synthesis and secretion of various biochemical structures serving as neurotransmitters. That's normally what the output is. What neurotransmitters are being affected by what stress, right? Of course. So the ventromedial hypothalamic nucleus, the VMH, has been assumed to play the role of controlling satiety, but newer findings have suggested it's the arcuate nucleus. This was now 10 years ago. Now we know the arcuate nucleus plays a major role. At what level? The NPY, the agouti neurons versus the POMC neurons. Right? They may serve as the authentic locus controlling bioenergetic and biofuel homeostasis. It's really important if you're studying obesity. The arcuate nucleus is anatomically positioned at the base of the brain's third ventricle, posterior to the medium eminence where the capillary endothelium lacks tight junctions. Therefore, this is very significant, results in a gap in the blood-brain barrier that can leak. Now, I put those in quotation marks because they're my terms for it. They can leak higher molecular mass signaling molecules and possibly even cellular structures like immune cells or pathogens, like bacterial cells, directly from general circulation. The hypothalamic arcuate nucleus is a critical mediator of energy and glucose homeostasis. We already know this. And there are two loci of that arc. The neurons, first order neurons, are the NPY and the agouti RP. Those are combined in one set of neurons. And then the POMC, right, the pro-opiomelanocortin, that we've talked a lot about now. Recall that the POMC is proteolytically processed into the adrenocorticotropic hormone, the alpha-melanocyte stimulating hormone, plus endorphins and ketones, lipotropins, the CLIP, and two other MSH isoforms. Stimulates the feeding via its anorexigenic binding to a particular receptor called the MC4. Okay, that's how the MSH works. One of the 5 known melanocortin receptors, so the number 4 seems to be a triggering one for anorexic responses. And it's downstream, and then everything downstream from those affected neurons. The second subtype are neurons that co-express the orexigenic peptides, neuropeptide Y, NPY, and the agouti-related peptide, GRP. And that results in the inhibition of the anorexic signaling. So you turn on one and you shut the other. So you get a double aspect, like I've been talking about in authentic biochemistry. So AGRP-NPY neurons synapse directly with POMC neurons, and they provide an inhibitory tone to that axis. And the AGRP itself is an inverse agonist at the MC4Rs, effectively antagonizing the effects of the alpha MSH, which normally will bind to that receptor, right? So you get the idea how this works, right? Both the AGRP-NPY and the POMC neurons are called first-order neurons, and they have axonal projections to other hypothalamic nuclei. Obviously, they don't work by themselves. And these are housing downstream second-order neurons as well. So the AGRP-NPY neurons project primarily then to the paraventricular nucleus, or the PVN of the hypothalamus, while the POMC neurons project more broadly, reaching other nuclei, such as the lateral hypothalamus, in addition to the PVN. Okay, so you see that they're not working at, it's not like when you get incident light on a smooth surface, and then you get reflection coming off at the same angle, right? A direct reflection. It's more like refraction because you're hitting a different layer. What's a different layer? The different level of neuronal tissue, right? So that's a metaphor. If you know anything about light absorption, reflection, refraction. All right. So collectively, the interplay between these orexigenic and anorexigenic neuronal subsystems I just told you about, which you've heard many times in these lectures, and their downstream effector signaling form the melanocortin signaling system. It is the activation of the hypothalamic melanocortin tone that regulates homeostasis. What homeostasis? Satiety versus the feeding response. That's the point we're making. Anorexic versus orexigenic. And thus has a handle on obesity. So here's a paper that I talked about in the audio podcast. You carry out a lipectomy now. That means removing lipid, right? You're not going to likely do this in humans, although there are surgeries where people remove adipose tissue, or surgeons remove adipose tissue from people, I should say. Anyways, this paper was published about 5 years ago, Journal of Applied Biomed. You can see its citation there. So when you carry out a lipectomy, what do you think happens? You decrease the amount of white adipose tissue, obviously, because that's what you're getting rid of. What that does, because leptin is generated there, you get less leptin. So what happens in the hypothalamus? Because we know that leptin is signaling there, right? This is a talk about hormones, after all. In the hypothalamus, you get then an increase in NPY, an increase in AGRP, because there's less adipose, and you get a complete drop in POMC, and yet this other genetic locus, CART. What's CART? This is interesting. CART stands for cocaine and amphetamine-related transcript. That's right, there is such a thing in the human brain and in the rat model, or mouse model. This is a pre-protein that is proteolytically processed to generate multiple biologically active peptides. Gee, that sounds like POMC, right? Remember, POMC has the opioid, the endogenous opiates like endorphins and ketones. This has a cocaine and amphetamine-related transcript. Interesting, right? Same locus, right? Same locus. All right. So all these peptides then play a role in appetite, energy balance, maintenance of body weight, and reward and addiction, and the stress response. So they are collinear with the POMC. Expression of a similar gene transcript in rodents is upregulated following administration of cocaine and amphetamine. That's why it's got that name. And mutations in that transcript, the CART, which obviously are going to make polypeptides, associate with obesity. All right. Anyways, when this happens, when you get a lipectomy and you run through this course of activities, these events, you ultimately get an increase in food intake and you get standard growth rate, right? Because you've removed the lipid. And so you're turning on then this whole orexigenic pathway, and you're tanking the anorexigenic. Right. Now, I want to remind you about this AMP kinase. Now, we're getting back into the subcellular location. We talked about hormones, so let's go back. Remember, the AMP kinase is also involved here. So when you get an increase in the ratio of AMP to ATP, because ATP levels drop. Remember, the way it works is ATP goes to ADP goes to AMP by removing phosphoryls successively, right? And the less ATP, the more AMP, signals to the cell that needs more energy, basically. So that turns on this enzyme called AMP kinase. AMP kinase does several things. One is it works through this tuberosis system, this PTSC2, TSC1, it's a dimer complex. And that blocks the Reb-GDP, G protein complex. Now, when it blocks that, Reb-GDP can't block this anabolic pathway called mTORC1. Because it can't block it, some P7056 kinase, 4,3BP1, and the sterile response element binding protein, that's what that stands for, turn on protein and lipid biosynthesis. Okay, but wait a minute. I thought that this was a stress, dietary deficiency, starvation mode, fasting mode. It is, but as I tell you all the time in authentic biochemistry, nothing is one way. There are no one-way streets here, right? There are no pathways that only go in one direction. Every time you initiate one pathway, you generate another equivalent pathway to start to pull back that response. That's what this is. But normally what AMP kinase does is it causes phosphorylate P raptor. Remember, it's a kinase. That's what that P stands for. And this other protein called 14.33, which has an interesting etymology, which I'll talk about sometime. That blocks directly mTORC. So while this allows mTORC to be de-repressed, this directly blocks it. So it's not letting mTORC just take back over and repeal the initial signature. No way. It just means that some of it is still working. That's what I'm trying to tell you. AMP kinase also directly turns on this PGC1 alpha. We talked a lot about this. With this in association with the peroxide proliferator activated receptor alpha, then directly turns on the gene. These transcription factors are the transcription of the genes involved in beta oxidation of fatty acids, and all the way down to ketogenesis, production of acetoacetate and beta hydroxybutyrate, which will be important in a moment here. This transcription factor system also turns on this FGF21, which induces lipolysis in the adipose tissue and also in, for example, skeletal muscle. So you get the fatty acids ready for the oxidation. This same transcription factor turns on gluconeogenesis, TORC. And this can work through this pathway or directly through the phosphorylation of PGC1 alpha down here in a combination with an interaction of HNF4, which I've talked about in previous lectures. So you get the idea what's going on here. You're getting ketogenesis because you're turning on in fasting state. You're getting ketogenesis and you're getting lipolysis of adipose tissue, but you're also getting some repeal of this even within the same system. Now, what is ketogenesis, right? So you get fatty acyl-CoAs that are beta oxidized. They can be beta oxidized all the way down to acetyl-CoA, that's what this is, right? Or they can be beta oxidized down to the C4 acetoacetyl-CoA. There's an enzyme which will convert acetyl-CoA, two of them, to acetoacetyl-CoA. That's called T2. You remember that. There's also this succinyl-CoA mediating reaction, okay, which is going to ultimately allow you to make HMG-CoA. And HMG-CoA is then going to be lysed to acetoacetate. Now this looks a lot like what? De novo cholesterologenesis, right? Except instead of using HMG-CoA and reducing it and going on to make cholesterol, you're going on and making ketone bodies. Now, another important thing here is that de novo cholesterologenesis is cytosolic, whereas ketogenesis in the early phases is mitochondrial. So you can keep the two pathways separated because there's an organelle separation. So anyways, what makes ketone body like acetoacetate? There's a ketolysis. There's this enzyme that uses succinyl-CoA, which is going to add another CoA to the system. Now you have acetoacetyl-CoA. The T2 reaction then can make two acetyl-CoAs, and they can be directly used for energy, right? Now you have acetyl-CoA, which can go directly into the TCA cycle, for example. There's also this acetoacetyl synthase or acetyl-CoA acetyl synthase. There's a direct conversion, just like down here. So you can take in now the cytoplasm, not the mitochondria of the extrahepatic cell. You can make acetyl-CoA. Remember I told you there are two ways to make acetyl-CoA in the cytoplasm. Remember the first one? ATP citrate biase, because there's plenty of NADH in that cell. It's a well-fed cell. And so rather than making just pure energy like in the liver, you start making lipids. I mean, fatty acid and cholesterol. All cells are capable of doing this. The direct effect of having a ratio of NADH over NAD is higher than unity. You block the dehydrogenase of the TCA cycle, build up citrate, it leaves the mitochondria. Here's another way in the periphery to generate acetyl-CoA. You generate acetyl-CoA. This is another source of fatty acid synthesis, cholesterologenesis, and then the intermediates there making farnesyl pyrophosphate, geranyl pyrophosphate for that farnesylation of proteins and geranyl of proteins you just saw in 3-4 slides back, which remember covalent modifications, which are going to alter entire alterations of gene expression from transcription to post-translational modifications. Okay. And see, all of this can be occurring because of a high-fat diet, right? Or because of obesity. So if you start to fast, some cells are fasting, and this is a good thing because then you're going to have a normal homeostatic control. But if you have an obesogenic system, people who are obese, all of these pathways become corrupted. And the ketogenesis is way low. And because of that, you don't have the normal pairing of lipid metabolism in the adipose and lipid metabolism, say, in the muscle tissue or the brain. This is why obesity is a global pathogenic state. It corrupts these normalizing lipid homeostatic pathways. And remember, we are oleaginous organisms, so once you corrupt lipid metabolism, having an excess of input, like an excess of lipid, being generated because of high carbohydrate diet or high protein diet, and then the sedentary lifestyle, you're going to corrupt all these pathways. So here's a paper published in EBioMedicine in 2015. I'm not switching gears. I'm just showing you how this interrelates down to the CNS. I'm showing you I can look at other literature, right? And we want to look at the literature, how it's affecting the brain, because this is where I like to go, because I like getting back to a neurological perspective when I can. Okay, what's this paper telling us? This is an MS type of document. Myelin that is talking about what? Removal of myelin, right? So myelin degeneration occurs during aging and in AD, Alzheimer's disease. Okay. White matter degeneration is actually predictive of cognitive failure. Okay. You can say cognitive alteration, that's why I left it blank. Usually it's failure over time. White matter is beneath the gray matter cortex. It's composed of bundles of axons, of course, which are also known as nerve fibers. And they connect neurons in different brain regions into functional circuits. Okay. It appears white in microscopy because it supplies electrical insulation myelin-covered axons. That's what it looks like in microscopy. It's formed by non-neuronal, of course, glial cells called oligodendrocytes. They wrap up to 150 layers of tightly compressed cell membrane around those axons, or insulating those really critical axons in the CNS. Myelin is essential for high-speed transmission of electrical impulses, and its damage can impair conduction, consequently sensory, motor, and cognitive function. So demyelination definitely is going to give you cognitive function and motor dysfunction. Cognitive dysfunction and motor dysfunction, right? Parkinson's disease, Alzheimer's disease, prefrontal dementia, aging, alcohol abuse, drug abuse, all these are going to lead down this way, right? Multiple sclerosis is one way of also getting demyelinated. They confer a similarity here. So the human brain continues to myelinate until at least the third decade of age. And the frontal regions of the cerebral cortex, which carry out higher level executive functions, are last to become myelinated. That's why it takes a long time for the human brain to mature. That's why younger people make more mistakes than older people. Then when people get really old, they start making the same mistakes that we were when they're younger, but in different ways, right? Because they've changed. They've gone through enough event ontologies that the way that they behave is different than when they were younger. Maybe. Who knows what they were like when they were younger? Often we don't know if it's our parents, for example. We might not even remember if it's us, if we have such a debilitating disease. Now, brain regions that myelinate late, like the frontal parietal tracts, the corpus callosum, the uncinate fasciculus, and the superior longitudinal fasciculus, are most vulnerable to breakdown and degeneration in Alzheimer's disease. Okay. It's just some disease etiology. Afferent targets of those fiber systems, like the hippocampus and the subiculum, are also high in white matter. Okay? Myelin breakdown manifests early in apolipoprotein epsilon 4 carriers. This particular genetic locus, which makes a person potentially predestined to get Alzheimer's. And that's a major genetic risk factor for AD, but certainly it's not necessary to get AD. The major risk factor for AD is age, and this is even more prevalent in females. And another major risk factor is age and obesity, comorbidities. The animal model studies have shown that a drop in mitochondrial oxygen consumption, CO2 release, and an increase in mitochondrial hydrogen peroxide are all associated with female brain senescence-linked Alzheimer's disease, and that the cytosolic phospholipase A2 sphingomyelinase pathways in function. Now we're going to show you that in a minute, what I mean by that. So ketones versus glucose shift in prodromal Alzheimer's. See how this is leading back now, right? Ketones. CNS bioenergetics are compromised in the Alzheimer's disease patient. Poor glucose utilization with elevated ketone body preference as biofuel is a metabolic phenotype characteristic of AD. Okay? Elevated ketones using as a biofuel in the CNS. A reduction in cerebral metabolic rate of glucose utilization, particularly in the entorhinal cortex and hippocampus, correlate with cognitive deficits over time and most accurately predict future cognitive decline. These are human studies in normal individuals as well as conversion to mild cognitive impairment, moving down the grade to AD, right? AD patients use a ratio of 2-to-1 glucose to alternative fuel, while controls are at or young controls exclusively use glucose with a ratio of 100 to 0, or maybe 100 to 1. Right? But nothing's ever 0. But this is what's been studied. Right? Lots of glucose being used when you're younger. As you age, you use more and more ketones. Why is that? Because you have more adipose, right? And because there's more of a lipogenic and a lipid homeostatic environment as you age. And that means after the third decade, most people don't think that they're old when they're 30, but already this process is occurring. I told you ketogenesis is good. It's good because it controls lipolysis. Lipolysis is being controlled. And if that's controlled, I mean the adipose tissue is being used. So having ketogenesis is good. Using ketone bodies in muscle is good. And even having ketone bodies during fasting or starvation or during severe dieting or during stress in the brain is good. But an excessive amount of ketogenesis in situ in the CNS. Problem here. So the shift from primary aerobic glycolysis to a ketogenic fatty acid beta oxidation pathway to white matter degeneration appears to be the key here. There's a paper published in Molecular Aspects of Medicine, 2011. There's your citation. So Alzheimer's disease is accompanied with a decrease in the expression and the activity of enzymes involved in mitochondrial bioenergetics and glucose metabolism, including pyruvate dehydrogenase and complex 4 cytochrome C oxidase. How interesting. In situ CNS ketogenesis and ceramide production, are they biomarkers for AD? Maybe. During starvation and certain reproductive states, as in gestation and lactation, massive increases in ketone bodies derived from liver metabolism of lipids across the blood-brain barrier, BBB, through the monocarboxylic acid transporter occur. Okay. Glucose transporters and MCT1 expression can also decline with age, and Alzheimer's disease leading to a compensatory response that shifts to the utilization of a brain-derived fatty acid to generate the ketones because glucose levels are diminishing. In the aging female central nervous system, myelin catabolism initiated by mitochondrial hydrogen peroxide activation of the phospholipase A2 acid sphingomyelinase pathway degrades myelin integrity while increasing in situ lipid droplet accumulation, fatty acid metabolism, and ketone body generation. Right? So you're breaking down myelin, okay? And in so doing, you're making the biofuel source to make ketone bodies because there's a deprivation of glucose. To get demyelination, to recover from, to compensate for, to complement the lack of glucose uptake because of the aging process, limiting the amount of glucose uptake because of the transporter systems and because of the lack of metabolic control in the glial cells, astrocytes in particular, versus the neurons. This is what it might look like. It's a paper published in 2015. It's the EBioMedicine paper. Here's your astrocyte, hydrogen peroxide, turning on phospholipase A2. That generates arachidonic acid, by the way, which can then be directly modified via the cyclooxygenase, lipoxygenase, or P450 oxygenase pathways, leading to highly reactive local hormones that cause inflammation and trigger inflammation. Okay? So that's now happening in the astrocyte. And here's your neuron, right? So you get this sphingomyelinase activation, but you get a breakdown of myelin, sphingomyelin. Then you get the sphingomyelinase making ceramide, which we already know is a harbinger, depending on the molecular species, of what? Programmed cell death, neurodegeneration, Alzheimer's disease, Parkinson's disease, prefrontal dementia, normal aging dementia, you see? But you also get the ketone bodies formed, right? So the ketone bodies then can be utilized, right? So you get this ketone bodies being made in the astrocyte and then being transferred to the neuron. So then you're able to get the carbon source to make ATP, and you're also making acetyl-CoA, of course, at the end product, right? So this is what the neuron starts living off of the carbon from the astrocyte, and therefore the demyelination carbon source, whatever that fatty happens to be. At the same time, enhancing inflammation of CNS. This is not a good thing. This is the source of a prodromal pathogenesis in AD. So if you think about bioactive sphingolipid metabolites in general, you get this sort of system. Sphingomyelin, you can get apoptosis and cell cycle arrest with phosphorylation, or sphingosine, with sphingosine. You can get phosphorylation to sphingosine 1-phosphate that causes cell motility, cell survival, cell proliferation, inflammation. None of those might be good, because if you get proliferation in the central nervous system, you can get a brain tumor, right? So there is a tight control over the amount of sphingosine versus sphingosine 1-phosphate. And we talked about this receptor-mediated responses for those various G protein-coupled receptors. We also know that there's this sphingosine 1-phosphate lyase, which will remove everything. And these products, ethanolamine 1-phosphate and formaldehyde, which actually can be toxic in the central nervous system, builds up. But these are end degradation products that can't get back into the pathway. Then you have ceramide synthase, something I talked a lot about. And it's going to be working in this direction, where ceramidase, making sphingosine, is going to work in the other direction. Remember, ceramidase is going to give you what? A fatty acid. And that fatty acid, depending, can go directly into ketogenesis and metabolic rate as well, or act as a signaling molecule for RAF formation. Yep. So ceramide is directly involved in apoptosis, cell cycle arrest for sure, and cellular senescence, that's aging. You know that sphingomyelinase can generate it. You know, de novo synthesis can generate it. Here you're seeing that ceramide synthase can generate it. You also have sphingomyelin synthase, which can pull ceramide back into synthesis of sphingomyelin. And then you have here the de novo pathway, right? We talked a lot about the SPT enzyme. Likewise, there are kinases, the ceramide kinase making ceramide 1-phosphate, and we have a phosphatase kicking it back to ceramide. So this is all in dynamic flux in the central nervous system. Ceramide 1-phosphate, kind of like sphingosine 1-phosphate, is associated with survival, proliferation, and with inflammation. Now, that's a problem, okay? Again, if it's the central nervous system. But it's also a problem if you have neurodegeneration and the whole lipid raft idea about aggregating and moving around potential receptors in the central nervous system. Right. I just want to put this together now with the whole system. Glucose is converted down to pyruvate in the mitochondria. Pyruvate, of course, pyruvate dehydrogenase. There's also this enzyme here, which will allow for the phosphorylation. But just look at pyruvate to acetyl-CoA to TCA cycle, making NADH, moving through the multiple complexes in the electron transport chain. It's not showing number 2 because that's an FADH-mediated response. So it's complexes 1, 3, and 4, of course. And these are basically all dehydrogenases, and you kick up ATP levels. Now, also remember that peripheral ketone bodies can be broken down. Acetoacetate can be broken down use of the succinyl-CoA reaction. Sux3-succinate is made. That's the Scott enzyme I showed you, making acetoacetyl-CoA, which can then directly enter into these pathways. One of the pathways, besides not shown here, that I mentioned to you is de novo fatty acid synthesis. So basically what happens is you're aging. This is down here now. This is the demyelination pattern, phospholipase A2, and then the removal of the sphingomyelin. You're making fatty acid. The fatty acid then can go through beta oxidation, coming directly from the myelin sheath, as for bioenergetics for the neuron. Remember, I'm telling you. You make the ketone bodies, acetoacetate, beta-hydroxybutyrate. Okay, it leaves the astrocyte. It goes through this transporter that's kicked up as you age. Both cells have this transporter. Comes into neurons, ketone bodies then are passed through this pathway down here, and you're back into making ATP in the neuron. Now, this is normally what happens during senescence, but you have to understand that in obesogenic system, this particular increase in lipid metabolism can be enhanced because of the excess amount of carbohydrate in the diet converting the carbohydrate to lipid. If you have just a high-fat diet in humans, it's not quite the same as in the murine models, because high-fat diet in humans tends to stop de novo fatty acid synthesis, even in the presence of lots of sugar in the diet, because that's how it works, feedback inhibition. So that's why the American Dietary Association said, well, wait a minute, maybe we shouldn't worry about fat, so much fat, lipid in the diet. We should be more concerned about carbohydrate in the diet. There's a lot of other reasons for that, and linked glycans and whatnot, right? Age proteins that we talked about in the past. But this metabolic grid in the central nervous system plays a key role in neurodegeneration in the central nervous system. I want to make sure you understand that. And I also am showing you how this is linked into hormonal regulation, endocrine hormonal regulation, and the immune response, right? Because of the inflammatory signaling. So we're going to stop there. This was Authentic Biochemistry, and this is actually May 6th of 2020. This is an Authentic Biochemistry podcast production by me, Dr. Dan Guerra, your host, and also the one who writes this and produces it and does all the literature research. Basically, the one person working on this. So there's my Facebook page. You'll find all of the ones that you're seeing here free. When you become my client, I will work directly with you. We will go into the scientific literature together, and we will find what you need to determine is necessary for your understanding of something from that scientific literature. And it's my job to deduce the data into evidence and evidence verification, ultimately where it's knowledge that can be used within the backdrop of all the other papers that have been published in given areas. This is the kind of demonstration I'm doing to you when I give you these free lectures. Okay. So let us finish this lecture off. Back down here. And I'm going to say, as I always do at the end of my lectures, bye for now.